So, we're just a couple of minutes over time, but I think we should just get started if that's okay. Please, I'd like to welcome you to the it's officially the rapid fire session, Epidemiology, Genetics, Public Health, Training and Education. I'm co-chairing. I'm Samantha Fraser-Bell.
And I'm Andrea Vincent.
Yep. So I just would like to start by saying the Royal Australian and New Zealand College of Ophthalmologists, members and participating guests wish to acknowledge the Aboriginal and Torres Strait Islander people of this country, sovereign custodians of country. We acknowledge their continuing presence and their contributions made to the college and the communities we work in. We will respect past elders and communities and will continue to live, work and interact in a respectful manner with present and future elders and communities. The RANZCO Reconciliation Action Plan reflects the messages of its First Nations engagement. Did you want to add in the New Zealand one? No. Okay. So just want to remind everyone that you can use the Congress app and then you can submit questions via the app. Please put your mobile phones on silent so as not to interrupt any of the speakers. And at the end, each speaker can speak for 5 minutes and then they'll have 2 minutes for questions. And we'll try and give you a warning if you're getting towards the 5 minutes.
Or throw something at you.
Yeah, one of those things. Maybe bang, because I don't think I've got a bell.
So, and if you're odd, please speak on this side. And if you're even, you can speak over there. So odd balls are here.
Yes.
Do we have our first speaker, Oliver Riley?
Did he come? No. Okay.
So we appear to be...
So even though we'll still call you even. Okay, yep, so good. So if we could please welcome Dr. Felicia Whittier-Putri. Her presentation title is Adherence and Barriers to the Recommended Diabetic Retinopathy Screening Guidelines in Pregnant Women with Pre-Gestational Diabetes in Metropolitan Melbourne. Dr. Felicia Whittier-Putri completed her PhD at the Centre for Eye Research Australia, University of Melbourne, focusing on diabetic retinopathy in pregnancy under the supervision of Associate Professor Lindell Lim, and she's in her first year of residency in the Department of Ophthalmology in Indonesia. Please welcome Felicia.
Good afternoon, everyone. Thank you very much for the opportunity to present my study today. So diabetic retinopathy may worsen during pregnancy in women with type 1 or type 2 diabetes. Up until now, there hasn't been an agreement upon guidelines for DR screening in pregnancy. In Australia, we have 2 sets of guidelines. The first one is from NHMRC, which recommends having at least 1 examination in early pregnancy. And the second one is from the Australasian Diabetes in Pregnancy Society, which my study protocol is based upon, that recommends having eye exams 3 monthly or once in each trimester. Unfortunately, we don't have any available data on the adherence rate to these recommendations, nor any knowledge of any barriers to these recommendations. And that's why my study aimed to fill these gaps. This survey study was a part of a bigger prospective cohort study of pregnant women with pre-existing type 1 or type 2 diabetes, and all participants in the cohort study were asked to complete a questionnaire using the compliance with annual diabetic eye exam survey that was validated to assess factors related to the eye screening adherence. Some modifications were performed to cover more frequent eye screening schedule during pregnancy. So in total, 134 participants completed our survey. We found that 85.1% adhered to the NHMRC, but only 20.9% adhere to the ADIPS guidelines. So let's start with adherence to the NHMRC guidelines, So this table shows data comparison between adherent and non-adherent patients. We can see that more pregnant women with type 2 diabetes were not adhered. Meanwhile, there was a higher rate of DR at baseline in those who adhere. Then when we look into the questionnaire, we found that the group who had better adherence were more likely to strongly agree that diabetes can damage the eye, that they feel that the examinations could find other problems apart from the DR, that they wanted to have regular eye exams and understood it's important. And finally, that they were happy with the care provided by the eye services. Furthermore, we usually thought that cost is a potential barrier to adherence, but this did not appear in our cohort, where those who did not adhere were more likely to disagree that eye exam cost too much compared to those who adhered. Instead, the belief that there was no treatment for DR was found to be the barriers to the non-adherence. So then how about the adherence to the ADIPS guidelines? Comparing between the two groups, we found similar findings that a higher rate of women with DR at baseline adhered to ADIPS recommendation, but those with previous child were more likely to not adhere. And from the questionnaire, we found that women with poor adherence were more likely to think that they could control their blood glucose well and their ocular health was not a priority. Then my next question was, what is their priority? I asked my participants to rank from 1 as the most important to 7 as the least important appointments that were required during pregnancy in regards to their diabetes. As expected, during pregnancy, the most important thing is their future baby. It can be seen here that obstetric and endocrinology appointments were cited as the most important appointments, followed by GP and diabetes educator. Eye exams were ranked second last priority, similar to dietitian. Fortunately, the majority of them still thought that their eyes are more important than their feet. So in conclusion, our study showed that adherence to this recommendation was still suboptimal. And we found some enablers and barriers to this adherence. And some of them were the knowledge about diabetes and diabetic retinopathy as enablers. And most important thing is that competing priorities are the most significant barriers. So the potential solution for this is that proactive efforts are required to educate women with this condition and the importance of the routine eye screening examinations. And there is an urgent need to integrate eye screening exams to antenatal care programs to prevent vision loss in this growing population. Thank you very much.
So does anyone have any questions? So I'll just ask a question. So I know you did some eye examinations as another part of your study in an obstetric sort of department.
Yep.
So has that been implemented as something that's now permanent, or was that just for your study?
Okay. So actually, I invited my patients to have the eye check in the optometrist clinic near the maternal hospital. So it takes some time and harder for them to come, although we actually provided help with the transportation, but still we actually cannot recruit all of the patients. And the problem is that about the time and about the problem with having another child who cannot and don't have anyone to take care of the other child, and so on and so forth. So therefore, we think that having the same like one-stop shop care, including the screening at the antenatal care, will be much easier for these patients.
Thank you.
Can I just ask, is the onus on, in this situation in Australia, is the onus on the patient to actually make their own appointment for the eye check rather than the service provider recalling them?
Yep. So currently, the patients need to make their appointment by themselves. And then the endocrinologists or diabetes educators usually will advocate and or just mention, you need to check your eyes. And that's all. They will not accommodate any more than that. Unfortunately, yes.
Thank you.
Okay, thank you very much.
So I'd like to introduce our next speaker, Dr. Sarah Crowe. Her title is The 4 Eyes Vision Kit, Trials of an Innovative Method of Vision Screening and Correcting Refractive Error in Remote and Disadvantaged Communities. And Dr. Crowe is based in Sydney, trained in ocular plastics, but has a doctorate in molecular biology from Oxford as a Rhodes Scholar. Welcome.
Thank you very much for that introduction, and nice to see you today. I would like to introduce the 4 Eyes Vision Kit. As you know, 1 billion people in the world are visually impaired simply because they can't access glasses. Thankfully, just recently, it seems to be coming to the fore in that yesterday, the Fred Hollows lecture by Professor Claire Gilbert, she highlighted the importance of this problem and how, as ophthalmologists, we are tasked at addressing it. The World Health Organization has labeled it a global health crisis, and the UN Resolution for Sustainability has labeled eye health care should be accessible to all by 2030. So this is a real problem. I encountered this problem firsthand when I was working in the Solomon Islands recently. There was very little in the way of medical or surgical treatment, but most of the people who walked into the clinic could have had better vision with glasses. Why were there no glasses? Well, it's pretty obvious, but the answers are there aren't any professionals to test for glasses. People can't travel to where there are professionals who can test them. And even if they got there, the glasses are too expensive. So I have to confess, I got a bee in my bonnet about this problem, came back to Australia determined to help solve it. I developed the 4 Eyes Vision Kit, which contains everything a basic health worker can use to test vision, test refractive error, and dispense customized, affordable glasses on the spot in remote and developing communities. It contains a visual acuity testing device, a vision testing wheel, which is basically a simplified 4-opter, adjustable stainless steel spectacle frames, which I'm wearing, a wide range of pre-cut optical lenses, including toric lenses, and a mobile application which ties everything together with simple step-by-step instructions. So this is the testing wheel. Each wheel here contains -4 to +4 lenses. It measures interpupillary distance here. It's portable, durable, and affordable. This is where you can adjust, This is where you can adjust the PD. And then there are add-on lenses which are added magnetically, and they can increase the range of power from -8.5 to +8.5 and correct astigmatism. So as you see here, there's the minus and plus add-on lenses, and +1, +1.5, and +2 toric lenses. And just to give you an example of how I've simplified the refractive process, these lenses are spherical equivalent. So they're not just toric lenses. So someone testing spherical refractive error, they can add the toric lens and they don't have to do weird calculations, because basically subjective refraction hasn't changed for 100 years. And it doesn't have to be as complicated as it is. What ties the whole thing together is the mobile application. And I've heard it mentioned that this is what they call low-tech to high-tech. We're using high-tech to low-tech. We're using high-tech to simplify the whole process so that anyone can do it. It's capable of working offline, so that where internet's not available, you can still use it. It has step-by-step visual instructions, so you don't require English or even much of an education to be able to follow the steps. It tests visual acuity. It tests refractive error. It has dispensing instructions for the glasses. And it records data so that we can audit those results and make sure people are getting decent results. But also it can inform health departments and professionals on what's needed out in these remote communities where we really don't know what's out there. It keeps track of the inventory so that we can replenish stock as needed. So nobody has more stock than they need, but they have a complete range of pre-cut lenses. And future capabilities, as digital health improves, we can scan for pediatric eye disease, diabetic eye disease, all kinds of things in the future can be added to it as things become available. So I'll just give you some photos of our last pilot project, which was in Hanuabada village in Papua New Guinea. And you can see that when local people, when local people are testing their neighbors, this is a critical part of it. When I try and refract someone in Papua New Guinea, they say, yes, doctor, because I'm a white old lady. But as soon as they get their neighbor up there, they're yabbering away in pidgin. And you know that they're getting, is it better one or two or whatever you're doing? They're getting the right answer. The testing wheel is on a tripod, so it can be on a table or on a tripod on the ground, so you can test kids. Here's a guy indicating the illiterate E signs, right, left, and so on. And this is the astigmatic correction, so that the client themselves, the patient, can turn that and determine where the axis of astigmatism is best for them. And in this trial, we had 3 lanes going at once. So I'll just describe a few details here. The spectacle frames are durable, and there's a few reasons why they solve a lot of problems. Firstly, the right lens and the left lens are the same, so we only need 1 set of pre-cut lenses. There's a protractor etched on the inside of the frame. Oops, I keep getting this wrong. It's etched on the inside of the frame, so the pre-cut toric lens has a little mark on it, and you can dial that in to correct astigmatism. It has very durable screwless hinges, which are impossible to break, pretty much. The optical center of the lens is the geometric center, so that the size and the PD can be adjusted with a crimping tool that changes the curvature of the nose bridge. Oh, and it comes in a number of trendy colors. You've got bronze, gold, black, and silver. We've carefully considered packaging in light of environment and shipping costs. So the lenses are kept in this little lolly roll sort of thing where they're all in there together. The frames can be shipped without being pre-bent, so that's 50 lenses, 50 spectacle frames right there. There's a bamboo box, which is compostable, and bags made out of recycled plastic. So this was our trial in Hanuabada village, which just before COVID hit us and shut us down. We trained 6 local people to use the system, and over 3 days, they tested 400 community members and dispensed 220 pairs of glasses before we ran out. And there was still a queue down the street, people who wanted testing. People who got the glasses gave it a 98% five-star rating for not only improvement in their vision, but improvement in their quality of life. It was significant. So the key features of the 4-Eyes Vision Kit are that it's affordable and transportable. It simplifies subjective refraction so that basic healthcare workers can use it in their communities. It involves on-the-spot dispensing, and it is community-based, so people are testing their own people. Please consider joining us. We need help with clinical validation, so if you do any studies or clinics in remote places, we can come along. If you can't come along, you could maybe consider making a tax-deductible donation. Thank you.
That's fascinating. We've probably only got time for one question, if anybody has one. Otherwise, yes? Just speak loudly.
What's going to make the biggest difference to visual deficiency in a developing country? Spectacles or cataract surgery?
Spectacles. Because spectacles, refractive error affects young people, people of working age. And the other thing is that even if you do cataract surgery, people who have cataract surgery still have refractive error. So in Papua New Guinea, I saw a lot of people who'd had cataract surgery and still couldn't see well enough to function. So, thank you, Hugh. So we've got the big wigs behind us. Now we've really got to take this problem seriously. The problem with it is it's simple, it's cheap, and it's not sexy. You know,
Can
it's
I
not.
just very quickly ask how much approximately? I mean, I know this is a well, it's not
Yeah.
really a prototype, but
Yeah.
have you costed out?
Yes,
per
yes,
pair?
we have. And the prices will come down as we start manufacturing. But at the moment, a pair of glasses cost about $7, and the testing wheel costs about $150.
Impressive.
Yeah.
Thank you.
Thank you very much. Thank you.
Thank you.
Thank you. So the next speaker we have is Dr. Amy Kalantari. Yep, you're there. Good. So Dr. Amy Kalantari completed her medical degree in 2019 through the University of Notre Dame, Fremantle, after completing, so that was her MD, sorry, after completing an undergraduate degree in psychology at the University of Western Australia. And she currently works as an unaccredited ophthalmology registrar with the Lions Outback Vision in Western Australia. So if you could please welcome Amy.
Thank you everyone for coming this afternoon. So I'll be talking about the use of telehealth during COVID-19 in Western Australia. So Western Australia is the largest state in the country, yet the capital city Perth, where that red dot is, remains the only tertiary level centre for ophthalmology services, despite being over 3,000 kilometres away from towns and remote communities in the Kimberley and Pilbara regions up here. Lions Outback Vision was established in 2010 to provide specialist care for patients living in remote areas. This was achieved through in-person outreach visits on the travelling vision van and telehealth, where patients presented to their local optometrist for evaluation and then had a video consultation with an ophthalmologist in Perth. Studies identified that this was cost-effective, achieved high patient satisfaction, and increased the uptake of Aboriginal and Torres Strait Islander patients by tenfold. Enter COVID-19, the pesky virus that halted in-person clinics and changed the way that the service provided care for patients living rurally. During the 3-month COVID-19 lockdown period, Outback Vision introduced 2 new teleophthalmology modalities to continue providing outreach care. Home tel involved a phone call to the patient in the comfort of their home. Hospital video involved a video consultation at the local hospital or nursing post. And Optom Vid was the pre-existing video consultation at the local optometrist. Registrars triaged referrals to one of the 3 modalities based on information provided in the referral, equipment required for evaluation, and the patient's location. If they had a complete workup conducted, then telephone was used, whereas hospital and optometry were used if further testing was required, such as slit lamp examination, biometry, OCT, or visual field testing. So we conducted a retrospective audit on how these 3 modalities were used during the 3-month lockdown period from the 23rd of March to the 5th of June 2020. So a total of 431 consultations were conducted during the 3-month lockdown period. As can be seen in this pie graph, there was a fairly equal distribution between the 3 types. We found that Aboriginal and Torres Strait Islander patients were significantly more likely to receive a home telephone consultation than a video consultation at the local optometrist. This was also true for elderly patients. Rurality was also important and was classified using the modified Monash model. Majority of the patients belonged to MM6 remote communities, and these patients were significantly more likely to receive a hospital video consultation compared with patients from extremely remote MM7 communities who were more likely to receive telephone consultations at home. Cataract was by far the most common diagnosis across all 3 modalities and was diagnosed significantly more from optometry consultations than the others. Other key conditions that it was used for were diabetic retinopathy, pterygium, and glaucoma. So there were 4 key outcomes from each telehealth consultation seen in this bar graph. And the most striking finding was that a significantly higher proportion of patients seen via optometry consultation were actually booked directly for surgery compared to telephone and hospital consultations. We also found that cataract was significantly more likely to be diagnosed in optometry consultations. So what does this all mean? Our study showed that telehealth provided a useful solution to the intra-regional travel restrictions that prevented in-person ophthalmology visits during the lockdown period. The two new modalities proved the most beneficial for Aboriginal and Torres Strait Islander patients, elderly patients, as well as those in extremely remote regions. This may be a reflection on greater access to telephone and local hospital or nursing post services as opposed to optometry services in the areas, which often actually relied on visiting optometry through Lions Outback Vision. Additionally, telephone consultations may have reduced the risk of COVID-19 exposure out in the community, and that may have been a more attractive option for some patients. Our study also supports the importance of collaborative care with optometrists to provide care for patients living rurally. And this is something that Outback Vision does on a daily basis. This may be particularly important for reducing surgical wait times as well for rural patients and reducing the wait for the wait. In order to do this, we have enabled comprehensive ocular examination and biometry with their local optometrists, and then have a remote ophthalmologist review the imaging and consent for cataract surgery via telehealth. This can be conducted safely with an experienced and well-supported optometrist. Since the pandemic, the Australian government has also recognised the importance of telehealth for remote communities and has actually included telephone consultations on the Medicare benefit schedule. And our study supports the ongoing use of telehealth for people living rurally beyond the duration of the pandemic. Thank you.
So there's no questions that have come through yet. I mean, I have one question that just how do you really diagnose cataract from a telephone conversation?
Yeah, so with those telephone ones, most of those patients had already been seen by their local optometrist. And because they had already had that examination with the optom, OCT and biometry done, the optoms are able to actually forward all the imaging and information on to us, and then we can have a chat with the patient, take a history, and actually go through the whole surgical and consent process with them over the phone. And then when they come to Perth or their local centre for the surgery, you can see them pre-op and, assess for those things, because that's a concern for some people is that, I think in an ideal situation, you'd see every patient pre-op look for those things that we look for. But in reality, I think there's lots of pros and cons for doing this.
So they were all triaged
Correct.
based on whether they had
Yes.
an examination
Yeah.
or imaging. So therefore, by default, the ones you were doing a telephone call
We'll mostly follow up
had
ones.
already been seen.
Yeah.
So you had that information.
Yes. Yeah. Otherwise, you just can't. You need clinical information to do it. Yeah.
Great.
Cool. Thank
Let's
you.
move on. Thank
Thank
you.
you. So our next speaker is Dr. Lisa Zhouting Zhu from Sarah. Her presentation is The Association of the Retinal Age Gap with Arterial Stiffness and Incident Cardiovascular Disease. Dr. Zhu is the current Senior Research Fellow within the Ophthalmic Epidemiology Department at Centre for Eye Research in Melbourne. Thank you.
Thank you. Thank you for your kind introduction. And I'm really honored to be here to give you a brief introduction about my project, Retinal Age, a novel biomarker for cardiovascular disease. So CVD is the #1 cause of death worldwide, with more than 17 million deaths yearly due to CVD. And the burden of the CVD is projected to increase to more than $1,000 billion by 2030. Luckily, 90% of the CVD can be prevented by addressing the modifiable risk factors, such as smoking and obesity. So it's very important to detect the CVD as early as possible to initiate the counseling and medicine. So far, a large number of the CVD risk scores have been proposed, such as a well-known Framingham risk score. However, these risk scores are limited by the lack of the end organ damage information, the information bias due to the questionnaire data, and also the involvement of the invasive procedures such as blood test. The heart and eye shares a lot in common in terms of genes, backgrounds, risk factors, and pathogenesis. And also the retina is highly vascular. Increasingly, the retina is considered as a window to the heart. So our research group was the first one to propose the concept of retinal age, which is a biological age based on the retinal imaging. There are two terms here. The first one is chronological age, which represents the passing time since the date of birth, while biological age indicates the health status of our body. So we leverage more than 10,000 fundus images from the healthy population to train and validate the artificial intelligence algorithm. The algorithm performed well with a median absolute error of 3.5 years. The smaller of the MAE is, the better accuracy the model is. So which is even performed better than the well-established aging biomarkers, such as epi clock based on the DNA methylation, brain age based on the neural imaging. And furthermore, the retinal imaging is fast, safe, cost-effective, and user-friendly. We further define the retina age gap, that is a gap between the retina age and the chronological age. A positive value of retina age represented the retina appeared older than the chronological age. We further find that the positive value of the retinal age gap increased the risk of 10 years mortality. So taken together, retina being the window to the heart, retina age gap being the reliable biomarker of accelerated aging, and accelerated aging being the most important risk factor for CVD, highlights the potential of the retinal age gap in predicting the CVD. So in the current project, we included more than 35,000 participants free of CVD at the baseline. Following almost 6 years later, we identified a total of 675 CVD events. We noted an L-shaped between the retina age gap and the risk of CVD, where the retina appeared 2.1 years older than the chronological age. The risk of CVD increased substantially. So in summary, retina is a unique window to the heart. The retina age is a more precise indicator of aging biomarker, even performed well over the well-established aging biomarkers. And retina age is non-invasive, cost-effective, fast, and safe way of measuring the aging. The retina age gap, which is a gap between retina age and the chronological age, can be used as a CVD risk stratification, thus enabling early prevention and intervention. Thanks for listening.
I know some days when I look in the mirror, I feel older than I really am, but does it vary at all? Can you have good days in your retina?
Yeah, it's. I think the concept of the biological age is that to let you know that whether you are in the accelerated aging and do something for your health. Yeah.
Sorry, any serious questions?
I think this is terrific. And about 20 years ago,
Yeah.
started
Yes.
to do something just like this,
Mhm.
but with clinical grading of retinal vasculature
Yeah. Yeah.
as a marker of cardiovascular disease.
Yes.
And we've been able to take it from sort of what was almost a kid's table sort of effort of trying to grade the arterial angles and straightness and stuff. Have it so it can actually be used as a proper system. This is terrific. Well done.
Thank you.
The only thing is, Hugh, with that, we used to get referred all these patients where GPs would ask us these kind of questions. And I go, can't you just measure the blood pressure and treat it? And like, don't wait. Or I always found that step to, because I think everyone should have their risk controlled. But I suppose that, to actually give an age is very
Yes.
meaningful to a patient.
Lewis.
Well done.
Thank you.
That's what I was asking.
Have you seen cases where somebody has improved their lifestyle factors and reduced
Slow down.
Yeah, it's a very interesting and great question. Actually, we got a collaboration with a research group, and we're trying to figure out whether the training, for example, exercise, will reverse the retinal age gap. And we're looking forward to the results, and I'll update it to you. Actually, we use the artificial intelligence algorithm to train the.
What did you train the AI on, I guess?
Yeah. I think it's a black box. Still, it's in the black box of the artificial intelligence. But still, we find out the heat map indicating that the artificial intelligence focusing on the retinal vessels. Yeah.
It's interesting. I was recently in LA, and the head of the department of Jules, Cedars-Sinai cardiology, nuclear imaging, said, we've decided that, people really should look at the blood vessels as a risk for cardiovascular. And I said, people have been doing that for years, Tien Wong.
Yeah,
And so here's these cardiologists who don't even know about it. Thank you.
Maybe in Australia.
Thank you.
So now I'd like to welcome Dr. Alexis C.C. Britton-Jones. She is a postdoctoral fellow in the Vision Optimisation Unit at the University of Melbourne and Retinal Gene Therapy at CERA. She completed her Bachelor of Optometry at the University of Auckland and her PhD at the University of Melbourne. Her current research foci are on inherited retinal diseases and ocular gene therapy. And her title of her presentation is The Victorian Evolution of Inherited Retinal Diseases Natural History Registry Venture Study, Characteristics of the 150 initially registered participants. Please welcome Dr. C.C. Britton-Jones.
Thank you. And thank you for attending this session. On behalf of our PIs and the Venture Consortium, I'm really excited to tell you about some of the work that we're doing to advance ocular genetics research and learn more about inherited retinal diseases from Melbourne, Australia. So until recently, as we all know, there were no treatments for our patients with inherited retinal diseases. But this field is rapidly changing. In fact, particularly over the last 5 years, since the approval of Luxturna for that one form of inherited retinal disease, there are now so many more DNA-based and RNA-based gene and cell therapy treatments that are being developed for inherited retinal diseases. But until we can get these treatments to every single one of our patients with an inherited retinal disease, there's still a lot more that we need to do in this area. And what we need to do now is, one, we need to have a better understanding of different inherited retinal diseases, learn more about their natural histories and why they progress differently. And two, we need to identify patients with inherited retinal diseases from the community and develop a database of patients that can be enrolled into future clinical trials. And that's what we're doing in our venture study, or the Victorian Evolution of IRD's Natural History Registry. So we enroll patients into our registry, either through referrals by their clinicians, or the patients contact the investigators. This enrollment then enables us to collect their retrospective clinical data to learn more about their conditions and understand how their condition has changed in the past. We then invite our participants to attend for a baseline examination so we can learn more about their inherited retinal disease. And this examination includes both assessments of retinal function, so visual acuity, perimetry, electrophysiology, retinal structure, so through imaging, and wherever possible, we help our participants with genetic testing and genetic counselling to confirm their molecular diagnosis. And we do this currently through sponsored programs and in close consultation with the Ocular Genetics Clinic at the Royal Victorian Eye and Ear Hospital. Of course, some participants aren't able to come in for an assessment, so we do register participants on our registry with only retrospective data as well if they're unable to. Using the information that we then collect, we can then enroll our participants into natural history studies, disease-specific sub-studies, as well as clinical trials. So where are we at now? Well, we've published the first database information of the first 150 registrants, and database has actually grown since then, but I will be just presenting on this data. About 60% of our registrants have rod-cone dystrophy, and this aligns with the estimate that rod-cone dystrophy phenotypes constitute about 60% of all inherited retinal diseases. About 30% have macular dystrophies, and the remaining other inherited retinal diseases. I'm not going to go into detail about the exact genotypes that our participants have, but I want to highlight that about 40% had a molecular diagnosis by the time that they are enrolled into our study. And the most common causative genes that our participants have are variants in RPGR, USH2A, CYP4V2, ABCA4, and choroideremia. And this indicates that a majority of our participants do have variants in genes that have emerging gene and cell therapy clinical trials. With this slide, I just want to highlight that most of our participants do have earlier stage disease. So we use visual acuity data at registration, which is available for about 3/4 of our registrants. So about 50% of our registrants have visual acuity better than 660 by the time they're enrolled into our registry. And about 30% have visual acuity equal to or better than 612. And so the key takeaway message is that to develop new treatments for inherited retinal diseases and to develop novel outcome measures, we need more natural history studies, and we need to learn more about the different inherited retinal diseases. And that's what we are trying to do in Venture. We're trying to collaborate with other Australasian registries to improve access to research and clinical trials for people with inherited retinal diseases. And really, it is our goal that every single patient with an inherited retinal disease knows about the research that's going on in this field. We accept referrals, so you can email your referrals to this email address. And really, I'd like to acknowledge the wonderful people that have all been a part of our work and who have collaborated with us. And finally, if you are not interested in hearing me answer the questions or have 5 minutes spare, please do scan this QR code and do the survey so that we can learn more about your practices in ocular genetics, and that will help us inform for future educational material and content. Thank you.
Lewis.
The amount of patients with ABCA4 disease in your neurologically diagnosed seems a lot lower than I would expect.
Yeah, it absolutely is.
Well, could I paraphrase that the number of CYP4V2 is overrepresented?
So yeah, definitely. So the aim of our registry has evolved sort of over the last 2 years. So we did start recruiting sort of during COVID in 2019 and 2020, and we all know how those 2 years have gone. But initially, Venture started because we wanted to grow a database for emerging clinical trials. And at the time, our group had a very strong interest in CYP4V2, as you'll hear very soon from Lewis's talk, sorry, I don't actually know if you're doing CYP4V2, but anyway, as you might have heard from some of the other talks that our group have done, as well as RPGR and choroideremia. As our database has grown, we have now really extended recruitment to include all other inherited retinal diseases. So even though now we do still have a strong interest in getting patients into clinical trials, we want all patients with inherited retinal diseases to know about the research that we're doing and have the opportunity to be involved in research if we can. And I do expect that as our database grows, we will get more of a representative cohort, but certainly the initial 150 participants do have a bit of, a referral bias for genes that are being evaluated in clinical trials.
Can I just ask, so you said 40% had a genetic diagnosis on enrolment. So those who didn't have a genetic diagnosis presumably had either been tested and didn't get a genetic diagnosis or had never been tested. Have you then subsequently tested them, and what's your strategy?
Yeah, we have. So we've been very lucky to have been working with sponsored testing programs over the last year, as well as having a close collaboration with the Royal Victorian Eye and Ear Hospital. So we have initiated or has started genetic screening for over 90% of the participants that's on our database at the moment. And that's either through NGS panels or it is through sort of collaborations or trying to chase up their genetic data if that's research level data. And we provide genetic counselling to every single one of the patients for whom we initiate genetic testing for. And that's one of the other goals of our group, is that we do believe that patients should have access to their genetic results, and we try and provide them with that service wherever we can. So hopefully we will get to keep doing it. But of course, as we all know, it depends on resources, funding, and various other factors.
Great, thank you.
Thanks, Andrea.
So I'd like to introduce our next speaker, Lewis Fry, RNA editing with CRISPR-Cas13 for the treatment of Usher syndrome. So Lewis has recently completed his DPhil at Oxford with Rob McLaren, and he is a New Zealander who has been poached by Australia. But we'll still listen to him. Like Russell Crowe.
Excellent. I'll just try and find.
Fred Hollows.
There we go.
You
Okay.
can keep Russell Crowe.
Thank you for having me, despite my trans-Tasman translocation. So yeah, my name is Lewis Fry, and I have spent a couple of years in Oxford working on gene editing therapies for inherited retinal diseases, including some of the ones that Cece has just spoken to you about. And in particular today, I'm going to talk about using Cas13 RNA editing for Usher syndrome. So Usher syndrome is Usher syndrome is a relatively common inherited retinal disease. Type 2 encompasses a non-progressive sensory neural hearing loss and a pigmentary retinopathy, and is commonly caused by mutations in the gene USH2A. And for many genetic disorders, AAV gene therapy has been investigated for supplementation, particularly in autosomal recessive disorders. However, one of the key limitations is that within these small and non-pathogenic viruses which are used for gene therapy, they can only take about 4.7 kilobases of genetic material. And we look at something like USH2A, it's 15.6 kilobases. So it has absolutely no hope of fitting within a single or even a triple AAV vector. And so other therapies are required. And so one of the approaches to doing that might be to edit the causative mutation directly. And so one of the common mutations, which I'm hoping Cece will identify in some of her registry studies, is this mutation which causes a stop mutation instead of a tryptophan in the 61st exon of USH2A. And essentially what that results in is the RNA is prematurely ended, and that results in its degradation, and any protein product is non-functional. And so one of the things that we could do to try and edit those mutations is to use something like CRISPR. And you may have heard of DNA editing with CRISPR-Cas9. Another technology is to use CRISPR-Cas13, and this focuses on using RNA editing. And so Cas13 editing involves using this deactivated Cas13 molecule which can bind to RNA molecules. And we can direct that by using a guide RNA, which is an antisense piece of RNA which can bind to the target molecule. And then added to that, you can use an adenosine deaminase protein domain. And this is a naturally expressing protein within the human body, and you can attach it to Cas13. And this catalyzes the conversion of adenosine to inosine, which you can see here. And inosine is read as a guanosine within the cellular translational machinery. And so essentially what you have here is an A to G conversion, which can correct G to A mutations. And all of this can be fitted within an AAV virus. So to do this for a mouse model, firstly, we needed to make a mouse model. And so that's what we did. We used CRISPR-Cas9 to insert a mutation of interest in the equivalent position within the mouse genome, and then characterize this mouse model. And what we could see is that the insertion of the stop codon resulted in the lack of expression of usherin at the connecting cilium. So you can see here we have, oops, I've gone one too back, but I'll go back. Hopefully we can see here. Got to get this cue card to work. We can see here we have usherin expression here at the connecting cilium and the photoreceptor in the wild type mouse, which is not present in the homozygous knock-in mutant. And this mouse also developed a sensory neural hearing deficit, and we're trying to characterize its retinal phenotype at the moment. But we pushed on to try and develop these therapies using a Cas13 editing in vitro. So in cells in a dish, we can use lots of different guide RNAs and lots of different protein configurations to try and find the most effective way of editing these nucleotides. We can screen a large amount of guide RNAs and find the most effective one. And then you can see here in a sequencing chromatogram, we have an A, which is here, which is almost completely converted to a G. So in the dish, we can get about an 80% correction. And then we can go forward to doing this in mice. And so I injected these adeno-associated viruses carrying the Cas13 molecules transsclerally and subretinally into mice that carried this genetic mutation. And as expected, we lose a lot of efficiency when we go from an in vitro scenario to in vivo, but what we can see is excitingly a 2% correction of the target mutation using the targeting vectors, which is not seen in our control conditions. And additionally, when we do that, we can see restoration of usherin protein at the correct location in the connecting cilium. And so this would be the first demonstration of the correction of USH2A in an in vivo model, and the first use of Cas13 RNA editing in vivo for retinal disease. So this is a really excellent result. So if we summarize what we have here is we've developed a technology further to use RNA editing in vivo for the correction of inherited retinal disorders. And this can be used to target many G to A mutations, which are particularly common throughout many retinal diseases, not only USH2A. And excitingly, this develops the use of RNA editing as a drug because, as you can imagine, RNA molecules are transient, unlike DNA. And so this allows us to titrate the amount of editing and potentially introduce safety signals to stop the editing if necessary. And then finally, all of this is AAV encodable within clinically approved vectors, and which could hopefully be translated to a gene therapy to continually express this RNA editing molecule within the photoreceptors and to continue to obtain editing levels, hopefully enough to restore vision, but we've got a long way to go there. So this is initial results in vivo, and excellent to present those. So thank you for your time, and welcome questions.
Hugh.
Changed mice, do they pass that on genetically, or do you have to do it for each generation?
Yeah, so it's done a... Unfortunately, at the moment, they can still see each other, which I was hoping that their retinal phenotype would be a bit more severe. But yes, no, it's done at an embryo level, and so then we now have a colony of mice, which we then breed together, and they pass on the mutation from generation to generation.
So, Lewis, if you corrected an RNA level here,
Yeah.
the newly, the transcribed wild-type protein, how long does that last? Like, you envisage you have to repeat this?
Yeah, it's an excellent question. So there's two parts to that, I would say. One is that as this is AAV expressed, you continually express the editing machinery. So for every new RNA molecule that is created, you continue to edit that, and that continues to translate to protein. And the second thing is usherin is a very large cytoskeletal, or like large structural protein, which is predicted to have a very slow turnover. And so the hope is that once you can create enough usherin, you'll be able to regenerate your photoreceptor outer segments. And that usherin should stay at the outer segment for an undefined but hopefully long period of time, predicted by its structural size, that we would hope would enable it to have a clinical effect. But it's an excellent question and one that needs to be answered.
Fred?
Yeah. Yeah, so this 2% result was 4 weeks post-injection, 6 weeks post-injection, yeah, 6 weeks post-injection of the RNA editor, and we haven't actually taken it out to a longer period of time. So in theory, you could build up, hopefully, a higher rate than 2%. But yes, as you say, we'll need to work out what amount is needed to restore vision. We know from studies in something like choroideremia, obviously a completely different scenario, that 5% should be enough to slow vision loss. So potentially 2% could be therapeutically relevant. Excellent.
Can I just ask why you chose that mutation? Because it's, I mean, you've got two. I don't know how many we've
it's
got.
present, I've got data on it, but present at about 5% of alleles worldwide, which is
But
not
is
bad.
that biased to?
Yeah, so it's about the third most common
It
mutation.
must be quite far
But
down in
the reason I chose it was it's a really good proof of principle. So one, it's a G2A mutation, it's not a deletion, and you can edit it with this. And the second is because it's a nonsense mutation, if you edit it, any restoration of protein you can see.
And is the phenotype always usher, I mean, with hearing, or is it can just sometimes be rod cone?
Yeah, you can have both a pigmentary
With
retinopathy
that.
or a syndromic presentation. Yeah.
Fascinating. Thank you. We should probably move on.
Yes. So please welcome Rachel Heath-Jeffrey. She is affiliated both with the Royal Victorian Eye and Ear Hospital and the Lions Eye Institute in Perth. And she's going to present her topic, which is, does a negative gene panel result in indicate a non-hereditary disease in patients with dominant retinitis pigmentosa? Please welcome Rachel.
Okay, so to date, 90 genes are known to cause retinitis pigmentosa, and 30 of these cause autosomal dominant RP. Some of these autosomal dominant RP genes are also associated with other conditions like Leber's congenital amaurosis, highlighted by the blue text, and cone rod dystrophy on the orange background, or a macular dystrophy on the blue background. The reported rates of resolving autosomal dominant RP pedigrees range from 47 to 70% in the literature. So the aims of this study were to determine the rate of positive gene testing in an Australian autosomal dominant RP cohort, to determine the rate of resolving inconclusive panels by additional interrogation and further copy number of variation testing, and lastly, to report the spectrum of disease causing genes in an Australian cohort. So we use two databases. The first is the Ward study, which is the Western Australian Retinal Degeneration Study. It has 760 patients with an inherited retinal disease, 330 of which have a rod cone dystrophy, so the percentage differing a little bit to CCs, but 43%, of which 106 have an autosomal dominant inheritance pattern. The Australian Inherited Retinal Disease Registry has over 4,000 participants with an IRD, of which over 3,000 have provided a DNA sample. In their cohort, over 1,700 have been diagnosed with a rod cone dystrophy, and what's interesting is that it's also 43%, so very similar to the Ward study. And over time, the registry has used different types of genetic analyses. So we had 54 probands that were recruited between 2003 and 2022. And of these, 4 declined genetic testing, which left us with 50. And of those 50, 4 were still awaiting results at the time of putting this together, which left us with 46. So that was 85%. So when we look at the baseline demographics of the patients of the pedigrees that were eligible, the mean number of generations with affected family members was 3.2. The mean number of affected members per family was 5.4. And the mean number of affected members examined per family was 2.2. With age, the mean age at symptom onset was 26. The mean age at the time of DNA sampling was 43. And importantly, the mean delay from DNA collection to reporting was 5.5 years, but this is changing, so at present it's closer to 12 months. So when we look at the gene panel results, so of the 46 probands with the genetic report, on the initial report, 27 were likely positive, 19 were inconclusive. And from these 19, 11 were further resolved with the additional interrogation by the Australian Inherited Retinal Disease Registry. So overall, 83% of these autosomal dominant RP families were able to have a genetic diagnosis, and 17% remained unsolved. So different methods of genetic testing were used throughout the study period, and they differ. So the first is an aspro-ophthalmic autosomal dominant RP microarray. It looks for known variants in known genes at the time of testing. The dates are listed. But the other 3 look for both known and novel variants. You can see that there's a very large number of genes within the MVL panel. And so it doesn't just include ocular genes, it also has hearing loss and those that cause neurological disease. In version 1 of the panel, it didn't have CNV analysis, which is copy number variation, but in versions 2 to 17, it did. So in summary, 59% on their first genetic report were likely positive, and a further 24% were resolved with additional interrogation, so 83% overall. When we look at the spectrum of genes within this cohort, two genes, Rho and PRPF31, comprised 33%. An additional 4 genes accounted for another 30%, and 8 genes accounted for another 20%, which left the 17% unsolved. So again, 59% of families returned a positive gene panel result. An additional 24 families had missed pathogenic variants initially. The failure rate, 17% of our families remained unsolved, and future whole exome and whole genome sequencing will hopefully help to improve this rate of diagnosis. So to conclude, half of those with an initial inconclusive gene panel result could be further interrogated like the use of copy number variation analysis, and a negative gene panel did not imply a non-genetic cause of a rod cone dystrophy. And lastly, additional analysis of intronic regions and future use of exome technology may further improve the rate of genetic diagnoses within the autosomal dominant RP cohort. And lastly, to acknowledge all the following bodies, but in particular the Australian Inherited Retinal Disease Registry. Thank you.
Are there any questions for Rachel? I have a question. So some of the ones that you may have done very early on with ASPA, have you retested them subsequently if they were negative? Is that your...
Only 3 were done with the ASPA and they all came back positive.
Ah, really? That's good.
So that had 100%, but I mean, it's not,
No.
it's not really, yeah.
And I recall, somebody may remember better than me, there was a meeting, Arvo or something, and it was in the middle of the night virtually. And there was a group that described a whole lot of unresolved dominant patients, and they'd found some complex intronic change. Does anybody recall that? Anyway, that might be worth, I will find it and let you know, might be worth pursuing. Ah, so they're doing mix, yeah. Is the ethnicity of your dominant unresolved families any... Mainly Caucasian.
Thank you.
Thank you. Right, our next speaker is Michael Dong. Hi, Michael. So Michael's title is RNA sequencing of human lens epithelial cells in post-vitrectomy cataract versus steroid-induced cataract. Michael is an intern at Alfred Health and is currently being supervised by Associate Professor Hall and Lim at Monash, and Sarah.
Great, thank you for the introduction and thank you for having me here. I'm very pleased today to talk to you about RNA sequencing of cataract. As you know, cataracts are still the leading cause of blindness worldwide, and whilst the vast majority of cases are age-related, we do know that cataract development is accelerated under certain conditions. A well-known side effect of corticosteroid therapy is the development of a posterior subcapsular cataract, and a common occurrence following vitrectomy surgery is the accelerated development of a typically dense nuclear sclerotic cataract. Whilst there are many theories as to why this occurs in both of these settings, there is no definitive underlying evidence as to what is the actual mechanism. Therefore, the aim of our study was to investigate the RNA sequencing differences in the genetic expression in different etiologies of cataract, specifically for steroid-induced and post-vitrectomy cataract. We performed a case control study investigating age-related, steroid-induced, and post-vitrectomy cataract. We performed sample collection by collecting the capsular rhexis at the time of cataract surgery by using a 30-gauge needle and submerging it immediately in a reagent that is able to stabilize the RNA at room temperature. Following this, we performed RNA purification, isolation, and quantification before sending the samples to the Australian Genome Research Facility for further library preparation and sequencing. We performed bioinformatics analysis using R and DE-seq2, and we defined differential gene expression as a up or down regulation of at least log 2-fold change greater than 1 and a p-value of less than 0.05. In this analysis, there was a total of 22 samples consisting of 2 age-related cataracts, 12 steroid-induced cataracts, and 8 post-vitrectomy cataracts. In total, we measured approximately 58,000 genes, and once removing the low abundance genes, we included 15,000 genes in the analysis. Principal component analysis allows us to look at the gross differences in gene expression at a very high level, and you can see that the steroid-induced cataracts and the post-vitrectomy groups, there's a clear difference there. And additionally, the age-related samples cluster within the post-vitrectomy group. In terms of differentially expressed genes, we identified a total of 30, 21 of which were downregulated and 9 of which were upregulated in the steroid-induced cataract when compared to post-vitrectomy cataract. In terms of upregulated genes, I'd just like to point out HECW1, which codes for protein ligase proteostasis, and also transforming factor alpha, which is part of the epidermal growth factor pathway. These are 2 genes that have been implicated in previous cataractogenesis studies in an in vitro setting. In terms of our downregulated genes, we were able to perform gene ontology analysis, which allows us to group individual genes into common biological pathways for more meaningful analysis. And interestingly, and by way of validating our results, we were able to demonstrate that in the steroid-induced cataract, there were downregulated immune pathways, including pathways in leukocyte-mediated immunity, innate and adaptive immunity, and as well as complement activation. But it's also important to note that whilst these pathways were downregulated, it may well be due to the steroid treatment rather than being causative for cataractogenesis. In conclusion, to our knowledge, this is the first RNA sequencing study to investigate the transcriptome of steroid-induced cataract and post-vitrectomy cataract. We were able to demonstrate with this methodology that different types of cataract exhibit different metabolic signatures. Steroid-induced cataract lens epithelial cells exhibited downregulated immune response pathways and upregulated genes that may be implicated in proteostasis and growth factor pathways. However, the main limitations of this study were the limited sample size, the low amount of RNA input, and batch-to-batch variation. Our findings also indicated that there is a possibility that the age-related cataracts are very closely related to post-vitrectomy cataract. However, this would warrant further studies with a larger sample size to investigate. Thank you for listening.
Hugh, please.
Yeah, no, this is a fascinating study. I mean, having spent too much time doing epidemiology of cataract, I mean, it's fascinating to see the genetic studies.
Genetics is just
everything.
everything.
That cause PSC, posterior subcapsular cataract, that are not steroid-related. Alcoholism is one. So, are you sure that your PSCs are actually steroid-induced and not related to the other risk factors?
That's a very good question. As part of this original methodology, we're actually hoping to compare uveitic as well as steroid-induced to sort of further delineate between the two. In terms of the steroid group, we actually specifically included patients who were on steroids with limited other sort of systemic disease, specifically without uveitis, so that we can compare between those two groups. But ultimately, as a preliminary study, we're only able to focus on sort of disease-specific sort of categories at this level, and we're unable to sort of delineate whether or not the posterior subcapsular cataract pathways are specifically due to steroid-induced, or if there's other factors in play, unfortunately. But I think further sort of subgroup analysis would be important with a larger sample size.
You could similarly argue that post-vitrectomy probably had steroids post-op, but actually the condition that led to them needing a vitrectomy, for example, diabetes, whatever, has other contributing.
But the post-vitrectomy nuclear sclerosis.
Sometimes you can get quite a posterior subcapsular component, can't you?
Not necessarily.
Maybe in Auckland.
And the post-vitrectomy are usually nucleosclerotic, which is really interesting and sort of bizarre.
Yeah, certainly in this group, all the cases in the post-vitrectomy were nucleosclerotic.
Thank you.
Thank you.
Thanks. And now I'd like to introduce Yong Min Lee, who's also known as Shane. He's a medical intern at the Royal Adelaide Hospital and Queen Elizabeth Hospital. He attended medical school at the University of Adelaide and graduated in 2021. I'd also like to point out he's a violinist and fingerstyle guitarist, performing as an artist in charitable events through the year, which I think is lovely and interesting. So his title is Ophthalmology Operation Note Encoding with Open Source Machine Learning and Natural Language Processing.
Thank you for the introduction. I didn't bring my instruments with me today, but today my talk will be on how we could use artificial intelligence to improve the accuracy of procedural coding. First, I'd like to thank my team for contribution, and to essentially appreciate this project, it's important to understand the current state of procedural and clinical coding. Now, this is a paper published in 2015 in the Annals of Surgery, and essentially involved about 30,000 patients. This study showed that upon review, about half of them, about 51%, required either a change in their procedural or their diagnostic codes. Now, the relatively high level of inaccuracy is pretty evident when you review the process. So once the surgeon decides to perform a particular type of operation, that generally corresponds to a particular code. In Australia, we use the Medicare benefit schedule. And then that code is then inputted into the hospital system to help generate a list of activity records for the department, ultimately useful for research and eventually financial reimbursement purposes. Now, as you can see, this is a long process and there's a lot of human input into this process as well, which is where the error source is from. Clinicians are often not trained. They are not aware of the significance of inaccurate coding. And unfortunately, in the public sector, with the high demand of patients to see, clinicians are often prioritized to see more patients rather than improve clinical accuracy. Then there are the codefactors, where these people are generally not trained into the specialty-specific terminology and unable to interpret data from this area. So this is where our team actually proposed the use of artificial intelligence, namely natural language processing models, to extract key information from an operation note, to identify what type of procedures was performed, and then to use machine learning to match that with the corresponding MBS code. So our method involved a manual encoding of 1,000 operation notes. We used a subset of that data to train our machine learning algorithm, and then we selected the best performing model and tested it on a smaller test data set. Now, the results were quite interesting, where first it demonstrated that our clinicians are performing at about a 33% accuracy. Our coders are performing at about 53% accuracy. There's a lot of room for improvement here, and so our machine learning algorithm demonstrated 88% accuracy for the 5 most common procedures. Then we started to look at this through a more financial analysis, and we demonstrated that our model was generating at about $923 per case, rather than this $1,072 per case, which it should have been. It was interesting to see here that our current coding is generating $997 per case though, and upon review we found that the reason for this is likely because the big surgery, the bulk primary procedure, is generally correct, but there's also a lot of errors in the primary procedures where over-coded for what was actually performed. So our approach is significant. It's basically, we're trying to revolutionize the process and approach to procedural coding. We would suggest a combined machine-led human approach to increase the level of undercoded activity, but also to decrease the level of overcoded activity. Having a better portrayal of department activity means that we can have better research purposes, we can have a more accurate financial reimbursement purposes. There's also other benefits, including benchmarking clinician performance and also surveillance of disease and procedures performed. But I guess the major benefit to this algorithm is that it is ultra time conserving, and it allows clinicians to focus on patient care rather than the clinical accuracy. I guess the future research in this line of work is to increase the level of accuracy for the rare type of procedures, which is where the model didn't perform as well in, and then to include multiple institutions, states, to make it a more generalized program, and ultimately to include other specialties with similar line of work to streamline the process of procedural coding as a hospital. Thank you very much.
So any questions from the floor? Otherwise, I'd just like to ask, so do you think there's much appetite to implement that if they're going to get $70 less or whatever it was per case?
I think the finance part is important, but for our purposes, is to increase the level of accuracy. So that is,
That's very true.
yes.
Yes.
Hi, very interesting talk. How do you ever go about imbuing works like this into an AMR, given the proprietary nature of all of them?
Sorry, what was it? IDMR?
How do you install this into an AMR?
Electronic.
Oh, EMR, right. So I guess this project will strive, especially in current times, because most medical documentation is an EMR form. In terms of implementing it, we will need to first have significant results like this, inform the public of the significance, and then to discuss with the hospital team whether we could implement this program into the EMR system per hospital basis.
Thank you.
Thank you.
Okay, and our final speaker today, I'm looking forward to this because we have some interaction, I believe, is Associate Professor Elaine Chong, who is based at the University of Melbourne, head of the Department of Ophthalmology at Melbourne Hospital and at the Eye and Ear. And the title is One Right Eye, a real-time virtual 3D model eye for personalised patient education. Thanks, Elaine.
Thank you. And just to add, I play no instruments. Maybe the only instrument is the phaco machine. Okay, so we're talking about one right eye. Okay, this is where we need to actually teach patients, what their eye condition is actually about. So can I get you to imagine that you are my patient and you have a shallow anterior chamber, or your child actually has cataracts, or your child has diabetes, but your dad has cataracts, and your mom has macular degeneration. And if you don't mind, we're going to add some drama. So when I clap my hands, the light is going to dim. I would like to introduce One Right Eye. This is a virtual eye model available online in real time. I can move the eye up, down, left, right, and zoom into the eye to explore the anatomy in greater detail. There is nothing quite like it at the moment. In fact, there are no eye models in clinic when I explain diseases to patients. So imagine you're my patient with shallow anterior chambers, or your child has diabetic eye disease, your dad has cataracts, or your mom has macular degeneration. So in a regular clinic without an eye model, I'll be explaining to you what a shallow anterior chamber means like this. Your cornea, which is the front part of the eye, meets the iris, which is the colored part of the eye, and this forms an angle. This is where the fluid drains out, and when shallow, the drainage is limited and the eye pressure can rise and cause glaucoma. So please give a rating out of 10, 10 being perfect. How much have you understood this? And write it down. So I've been using One Right Eye daily when I'm seeing patients, and this is how I do it. This is the cornea, which is the front part of the eye. It meets the iris, which is the colored part of the eye, forming an angle here. Fluid drains out through the angle. And look at the iris. When the iris is shallow, see how it becomes shallower? The fluid doesn't drain out properly. The pressure hence builds up, causing glaucoma. So I use the exact same words in my explanation. Please score this out of 10, 10 being perfect. So I hear you ask, what is glaucoma, doctor? Well, glaucoma is a disease of the optic nerve, and with high eye pressure, the nerve dies away and it is not reversible. This is why prevention is important. So this is the healthy nerve layer, and with high eye pressure, the nerve dies away and atrophies. This part atrophies, which causes vision loss. Now, your child has diabetic eye disease. And these are usually young people with full-time jobs. Diabetes can be mild, where there's only a few bleeding spots, moderate, where there's a few more bleeding spots, severe where there's vascular and more bleeding spots, or it can become progressively worse and become proliferative with all these new blood vessels forming and also bleeding into the eye. And all these depends on sugar control. It is therefore why it's so important that we monitor change over time to prevent these advanced changes when vision is affected and laser treatment needs to be started. Diabetes can also cause the back of the eye, which is this middle part of the eye, which is the important part of the eye where center vision is affected. And if this happens, you require regular injections into the eyes every month to treat this. This is why optimizing and controlling the sugars and attending clinic follow-up to prevent worsening is so important. Now, your dad has cataracts, so give this a score 2. So without an eye model, a cataract is a cloudy lens inside your eye. It is blocking the light entering your eye, and we should take it out of an operation. This is how I explain it in clinic with the one right eye. You have a cataract, and if we split the eye in half, this is the natural lens of your eye, and light can enter through this lens and get it focused to the back of the eye. As you get older, it becomes cloudy, and this becomes a cataract. It blocks the light entering your eye, resulting in decreased vision. And so if we remove the cataract like so and put in a transparent lens, light can now re-enter the eye and you see better. This is also the time where I can put a lens in your eye, which will allow you to see better without glasses. Now, your mom has macular degeneration. So macular degeneration just starts out with a few yellow dots at the back of the eye, which is the retina. And this doesn't affect your vision, but it can suddenly change to advanced macular degeneration, such as wet macular degeneration, where the middle part of the eye, which is the important part of the eye, important for central vision, is affected. So it bleeds and fluid leaks out into the retina. Now, you will need injections to control this bleeding and fluid leakage monthly to preserve vision.
So the problem is that we have the lights on, please. Thank you. When I clap. So the problem is that there's no eyeball models in clinic. And this is 2022. We should really have a better system. And if you're lucky we had an eyeball model, parts of it is missing and you can't find the cornea or the lens. So which is why we need to update our service to patients, educate our patients well. So we need to improve patient understanding, improve follow-up, have better patient compliance back to clinic with better treatment compliance, decrease chair time, and this should increase our consult efficiency. Now, if you don't mind, please scan this because this is actually a survey that I would really appreciate if you can fill in for me. Yours, do you want to come up and have a chat about this? So yours is an ophthalmology registrar in Western Australia. And the team is me, yours, and our ORE design team. So the QR code is still there. If you don't mind, just fill this in for me. Do you want to explain the survey?
Yeah, so the purpose of the survey is really to get some initial feedback as to, if any of you think this has some real world clinical utility. And also to just understand if anyone has any interest in being involved as a future tester of the product to see how it fits in the real world. This product right now is in its early stages of development, and if anyone's interested to be involved as we develop it further, your input would really change the direction of product development as we continue, depending on how it works in the real world. Ultimately, this was born from our own frustrations in clinic, trying to find spare parts of the plastic eye model. And we hope that you're able to provide some input that could direct the future development.
Yeah, so if there's any questions, please feel free to email me here. And if you want to be part of the beta testing, which is free for you, please enter this survey, put in your details, and we'll contact you for some free beta testing.
Anyone who's involved
you don't have
in...
New Zealand on your
your
Oh,
survey.
I'm so sorry.
Yes, come on.
I'll take responsibility.
Get a hero for that.
Yeah, I'll just say that, you know, anyone who's involved will absolutely have some benefit in the final product and you know,
Down the track.
Yeah, down the track.
Excellent.
Thank you.
Are there any questions before we've got a little bit? We're right on time, really, but if anybody does have any further questions.
Yep, go ahead. No.
Have you got pre-recorded ones that you leave outside?
Yeah, that's definitely doable. I mean, I pre-recorded this because I didn't want to fight with the internet here. But it's actually very easy to customize all the videos. So this is actually real time. So I can move it and I can show you, you know, if you have macular degeneration with a cataract, I'll show you at the same time your cataract and your diabetic macular edema right at the same time. So you can have multiple issues and it can be, you know, specifically tailored to that patient. I can also annotate on there's an annotation tool where I can annotate. And the future iterations would actually be telehealth compatible. So, you know, you could actually be in Darwin or whatever. I've tried to tell Angus to do this, but anyway. So you could be in Darwin, FaceTiming or remote in. The system will sync together. So whatever I'm doing on my screen, the patient can see on their screen and they can annotate at the same time. They can annotate back with a different color and I can see it as well. So, yeah.
Those are certainly the kinds of feedback or comments or features that you'd like to see that would be important. And, you know, having the ability to record your own voice for your practice to make it seamless with, you know, seeing the patient in your consult room and have your voice narrate the video is also something we could think about.
Which is easily done, like what I've shown you just now. Thank you very much.
Thank you.
Thank
Thanks.
you. So thank you all for coming and for a range of really fascinating talks here today. I think you can rate the session on the app.
Maybe.
Otherwise, have a wonderful evening. Thank you.
Thank you.